Welcome to section 15.7, where we're going to discuss the interactions between the parasympathetic and sympathetic divisions. Here I wanted to do a quick concept overview since it's been a minute since we reviewed some of the anatomy and structure of both parasympathetic and sympathetic divisions. So first we'll start off with the parasympathetic division. That is our rest and digest division. We also call it the craniosacral division because we have these nerves originating from the cranial nerves and the sacral region S2 to S4. We also know that the preganglionic neuron is going to be long and myelinated, whereas the postganglionic neuron is going to be short and unmyelinated. We do have some terminal ganglia lying near the target organ, or intramural ganglia that are located within the organ itself. The neurotransmitters released from the preganglionic neuron is acetylcholine, and that is going to bind to our cholinergic receptors, specifically the nicotinic receptors here. And that postganglionic neuron is going to release acetylcholine as well, and that is going to bind to our cholinergic receptors, specifically the muscarinic receptors on that target cell. Some of the effects that would help us rest and digest or return the body to homeostasis would be pupil constriction, increased motility and activity of our digestive system, our storage of fuel molecules within the liver, such as glycogen, and stimulates secretion of our lacrimal gland or our tear gland, our nasal and digestive system glands, such as the salivary glands, and lastly, decreasing our heart rate and causing bronchoconstriction within the lungs. Now for our parasympathetic division review, this is our fight or flight division. We are going to see that its preganglionic neuron is going to be short and highly branched. It'll also be myelinated, whereas its postganglionic neuron is unmyelinated, is long, and is not very branched. We are going to see that the fibers originate from T1 to L2, so we also call this our thoracolumbar system. And our ganglia are located near the vertebrae. So this is our sympathetic trunk within here. We also have some prevertebral ganglia that are located anterior to this, but still lie close to the vertebral column as well. You can see them pictured over here. Remember, they were lying anterior to that aorta. When we look at the neurotransmitters and receptors, we are going to see in one condition that preganglionic neuron, well, actually in both conditions, we're going to have it release acetylcholine and bind to cholinergic receptors, specifically the nicotinic receptors in both of these pathways here. And the postganglionic neurons can differ and either secrete acetylcholine to muscarinic receptors, or it can release norepinephrine to adrenergic receptors. And fulfilling the fight or flight response, we will have the effects listed here, such as pupil dilation. We will stimulate our adrenal medulla to secrete epinephrine and norepinephrine to prolong these sympathetic effects. We're going to decrease motility and other activities of our digestive system. We want to release our fuel molecules from our liver and our adipose cells and into the blood so they can be utilized. And we're increasing secretions of our sweat glands, our contraction of the erector pili muscle to cause goosebumps. We also have vasoconstriction of most of our blood vessels, but vasodilation of our coronary arteries and the arteries to our skeletal muscle so that they can function efficiently, because we are going to increase that heart rate of our muscle and the force of contraction, and also cause bronchodilation within the lungs. Now let's move into some new material. We'll see that most of our effectors are innervated by both divisions of our autonomic nervous system, and both will continuously release neurotransmitters, generating what's known as autonomic tone. You can define autonomic tone as being just the general activity rate of both the sympathetic and parasympathetic aspects of the system. And in some instances, we're going to see that only one division controls the status of an effector. So a great example of this would be our blood vessels. They are in a state of partial constriction due to sympathetic tone. Specific to our blood vessels, we call this vasomotor tone. The vessel is going to constrict, or that lumen will become smaller, if our sympathetic activity is increasing. Let's say you start exercising, but then as you stop exercising and your body comes back to homeostasis, that vessel is going to dilate because that sympathetic activity is decreasing. And remember, dilation is when that lumen increases. And for our vagal tone, we are going to continually decrease our heart rate. And remember, our vagus nerve, which is referenced to in terming this vagal tone, has parasympathetic fibers moving through it. I mentioned at the beginning of the chapter that your ANS has something called dual innervation, meaning that most of our organs are going to receive input from both sympathetic and parasympathetic divisions, and they're going to have antagonistic or cooperative effects. So let's talk about those antagonistic effects. This is common where we have the two divisions oppose one another. We can look at the control of heart rate, where our parasympathetic system is going to slow our heart rate and sympathetic is going to increase our heart rate. Our cardiac cells actually have both cholinergic and adrenergic receptors to allow this to take place. When we look at the muscular activity of our GI tract, we know parasympathetic is going to increase GI motility, whereas sympathetic is going to decrease motility. And our GI smooth muscles are also going to have both types of receptors. When we look at our pupil diameters control within the iris of the eye, our parasympathetic activity will decrease the pupil diameter, resulting in pupil constriction, whereas sympathetic activity is going to dilate the pupil, making it wider, allowing more light into the eye. And our parasympathetic fibers are going to innervate those circular iris muscles. Remember that picture from our previous slide? This one over here, where we're going to see those parasympathetic fibers innervating these circular iris muscles and the sympathetic fibers innervating this radial iris muscle. And now for our cooperative effects. We see these effects when our parasympathetic and sympathetic stimulation have different effects that are part of the overall response. So we can see this in the male sexual function, where the penis erection is due to our parasympathetic activity and ejaculation is due to the sympathetic activity. So they're working together in order to have the male sexual function perform. And when we see opposing effects, they can be achieved without dual innervation. So we talked about our blood vessels constricting with increased sympathetic activity and dilating with decreased sympathetic activity. Some examples of effectors that are innervated only by the sympathetic division would be our sweat glands in the trunk, our erector pili muscles in the skin, and the adrenal medulla neurosecretory cells. Now for a clinical view on Raynaud's syndrome. This can be caused if somebody is exposed to cold, stress, or have changes in their relative temperature. All of a sudden we have constriction of small arteries to the digits, which results in loss of normal hue of the distal skin that you could see pictured over here. We have that white pigmentation due to lack of blood flow. When someone is suffering from this, they have a hard time circulating their blood through their vessels and doesn't get back to the heart properly, and can be due to this exaggerated local sympathetic response that you have within your body. So in this first illustration, we see that we are having that lack of blood flow because of that circulation not taking place properly. In our second photo here, we see that the fingertips are more blue, and that's because we are lacking oxygen to reach the tissues here in the distal digits. And lastly, the hand turns red when that blood flow returns. And this is more common in women, especially within their teens or 20s, but symptoms tend to improve over time, usually by the time they're in their 30s or 40s. There are some other symptoms that accompany this. It could be pain, heart palpitations, sweating, or difficulty breathing. And if someone is prone to this condition or syndrome, then they should do things like live in warmer climates, or if they do have exposure to cold, running their hands in hot water or holding onto one of those warm packets throughout the winter season to make sure that their condition does not become exacerbated. Another clinical view here on drugs that affect pupil size. Within our parasympathetic division, we know that we're going to stimulate pupil constriction, and sympathetic division will stimulate dilation. Drugs that can also affect our pupil size would be tropicamide, which blocks the muscarinic receptors and causes pupil dilation. We also know of some illicit drugs such as cocaine, amphetamines, LSD, PCP that will also dilate the pupils.